Okay, folks, we're going to do a recap, maybe clarification of enzyme inhibition as a supplement to what we went through in class. So this is just the enzyme inhibition section of the lecture. There are various different reasons why enzyme inhibition might happen. It is a normal biochemical process, very often used within cells under normal circumstances to regulate enzyme activity. Because as we discussed in chapter 6, you don't want enzymes active usually all the time, just under certain circumstances. And one of the ways that this can be managed is by having enzyme inhibitors present, which can then be, removed or degraded when the enzyme is needed to work. Enzyme inhibitors can also be a very attractive target for drug designers. If an enzyme is known to cause some sort of a pathology, then it can be a target for drug designers to look for something that is then going to inhibit the enzyme. There's 4 different types of inhibition. Most inhibition is reversible inhibition, and the 3 cartoons that are shown here represent that. And then there is also irreversible or suicide inhibition, which we'll go through a brief overview of. These little cartoons here are intended to sort of give you a way of maybe, visualizing or understanding the concepts of the different types of inhibition a little bit more clearly. I won't spend a ton of time on them. Your competitive inhibitor here is where the enzyme will bind the inhibitor in the active site in place of the substrate. So it is a mutually exclusive arrangement here. The enzyme will bind either the substrate or the inhibitor. On the far side over here, uncompetitive inhibition, this is where the inhibitor will only bind the enzyme after the substrate has already bound. So where this is affecting binding of the substrate, this is affecting kcat, right? This is affecting converting that enzyme substrate complex into product. And then non-competitive inhibition, this is sometimes called mixed inhibition. This is a little bit different, and this is where your inhibitor specifically is going to bind to a place on the enzyme that is different to the active site, and it can bind either before or after substrate binding. This is showing some Lineweaver-Burke plots, right? You will definitely be expected to be able to articulate what are the effects on the kinetics of different types of inhibition. Some of them are going to affect Km, some will affect Vmax, some will affect both. Understanding why these different things, these different parameters are affected is going to help you. I mean, sure, a lot of you are just going to remember it by rote, but if you understand why the different parameters are affected, that's certainly going to help with retention of the information. Before we get into the specifics of those, we will briefly talk about suicide inhibition here. So a suicide inhibitor, this is going to be one that is covalently bound irreversibly and permanently to the active site of the enzyme. So typically it will look somewhat like the substrate. And you can see here in the example shown, thymidylase synthase is the enzyme that we are going to consider. The substrate is DUMP, which is a precursor to DTMP, and this is involved in nucleotide synthesis, right? So your inhibitor looks awfully like the substrate. The process of converting this is essentially adding a methyl group. You can see in red here, your methyl group is indicated in the product. What happens with the inhibitor is, because of this highly electronegative fluorine atom, when this inhibitor goes into the active site, this is so electronegative that it's going to pause the conversion within the active site, and the methyl group cannot be added because the attraction between this and electronegative fluorine and the enzyme active site is too strong. And so this will permanently inactivate the enzyme that it binds to. On to competitive inhibition. So your competitive inhibitor, as we mentioned, this is where your inhibitor is binding to the active site. So it'll bind instead of the substrate. It'll be one or the other. This is reversible, so it's not going to be a covalent bond. And it does mean that when your inhibitor binds, it affects substrate binding. So that should immediately get us thinking, oh, then does it affect Km? And in fact, yes, it does. But also what it means is that because this is just pure competition, whatever there's more of is going to win the fight here. So even if you have inhibition that is pretty effective, if you increase the substrate concentration, you're going to overcome that inhibition. And in fact, if we look at the kinetics of this, so without inhibitor here being shown in the left-hand plot, so the more the steeper slope here, and then with inhibitor in the red, the Km is what's primarily being affected here, because you are affecting the binding of the substrate to the enzyme, and that's kind of what Km is looking at. Now, as you increase the substrate concentration though, once the substrate's bound, there is no effect on the catalytic turnover, right, on the Kcat. So what that means is, as you increase the substrate concentration, you can overcome that, and your Vmax will remain the same in the presence of this inhibitor. Okay? And if we were to look at this information in the form of a Lineweaver-Burke plot, this is what we see. So again, the green or the lower slope line here showing the uninhibited enzyme, the steeper slope, the red line showing in the presence of inhibitor, and you can see that the line sort of like pivoting around the 1 over Vmax here, indicating that there is no change to the Vmax of the enzyme. However, where it's crossing the x-axis is different, and this is representing the increase in Km. Transition state analogs, I've mentioned this already, that they will often make better inhibitors than substrate analogs, and in fact transition state analogs are often sought after as targets by drug designers. So let's remember that transition states correspond to high energy structure where the bonds are in the process of breaking and forming, right? And so here we've got phosphoglycohydroxamate, and this resembles the proposed transition state in this reaction of glyceraldehyde 3-phosphate to dihydroxyacetone phosphate. And in fact, this intermediate is a proposed intermediate based on what is a known inhibitor of this reaction. Okay, so very often the intermediates, because it's hard to tell the structure of an intermediate, right? You can look at the substrate and you can look at the product, but it's kind of hard to figure out what's going on within the enzyme. But this was determined based on the fact that it was observed experimentally that this product here, or this compound here, your phosphoglycohydroxamate, has a binding affinity of around about 300 times as much as your glyceraldehyde 3-phosphate. So it's a really, really strong binding there. And because it binds so much, right, then it's not going to it's not going to just like, release it. It's pretty effective at sort of subverting what it is that the enzyme was initially supposed to do. So these are used frequently, or at least analogs of the transition state, if the transition state is known, or it can be maybe hypothesized based on what the structures of your substrate and product are known to be, because they will bind more strongly. So uncompetitive inhibition, this is a slightly different order than it was covered in the original videos that are posted on Brightspace. Uncompetitive inhibition, this is where your inhibitor will bind to the enzyme after the substrate has already bound. This is somewhat rare, but it does occur in some multi-substrate reactions. And the effects on this is that it is observed to reduce the Vmax and Km both by roughly the same amount. Okay, so the key here is binding after the substrate binds. So it's affecting the Kcat, okay? The reason that it will also affect the Km is because the loss of functional enzyme substrate complexes here is going to lower the apparent value of the K2 in this expression. So, like I said, this is kind of rare, but it does occur sometimes. Challenge for you, should you choose to accept it. Sketch a Michaelis-Menten plot showing an enzyme in the presence and absence of an uncompetitive inhibitor. Okay, so knowing that your Vmax and Km are going to be affected, how will that look? And then finally, non-competitive inhibitors. This is the area specifically that I wanted to clarify, okay? This sounds like it's really easy to confuse with uncompetitive inhibitors, but it is not the same. These are sometimes called mixed inhibitors. The binding happens at a site that is not the active site, so somewhere else on the enzyme. That's what this cartoon here is showing. So it's binding, way far away from the active site. So it doesn't necessarily alter substrate binding to the inhibitor, but it might. The initial representations of this, and a lot of the representations, if you were to Google image the kinetic parameters of non-competitive inhibition, a lot of times they state that it doesn't affect substrate binding, therefore Km is going to be unchanged. But in fact, it does sometimes affect substrate binding. So sometimes the Km does increase. And so this sort of like vague, sometimes it does, sometimes it doesn't, that is what I want to sort of include here. It is not as simple. In the situations, the very rare situations where this happens, where your enzyme or where your inhibitor is binding to a different site and there is no effect on substrate binding and only Kcat is impacted. So impacting Kcat is going to decrease Vmax, okay? So in those instances, that's referred to as like pure non-competitive inhibition, but usually there will be some sort of an effect on Km. It might be increased, but sometimes it's decreased. So what we end up seeing is something like this, okay? So this is an example of what it might look like. It doesn't completely prevent substrate binding, as would happen in the case of competitive inhibition, right? But it may affect it. It may decrease it somewhat. But unlike competitive inhibition, you're also going to have a significant effect on the Kcat. The ability of the enzyme, even when the substrate is bound, to form that catalysis or to do that catalysis is going to be impacted, and that's what decreases the Vmax, as well as affecting the Km. So this will be your challenge for this part. Can you draw a Lineweaver-Burke plot that corresponds to the data that's shown here? So unlike what you saw for non-competitive inhibition on the first slide, right, that simplistic kind of graph that was showing sort of the three lines are coming off of the negative 1/Km point, I want you to try and draw me a Lineweaver-Burke that shows what you see on the screen here. So transform this. So it is affecting both Km and Vmax. Let's see, allosteric regulation. This is a very common type of regulation. It is pretty common in oligomeric enzymes, and a lot of enzymes are oligomeric, anywhere from like, I don't know, I've seen it reported that 10% or even up to 50% of enzymes might be oligomeric. So if it's oligomeric, it means that there's more than one active site. And the allosteric regulation, so it's not being referred to simply as inhibition here, right? Because sometimes the allosteric regulation could increase the activity of the enzyme. And so an example of that might be phosphofructokinase, which is inhibited by a downstream product of this pathway. So this pathway, for those of you who don't know, is glycolysis, right? Now, we haven't gone all the way to the end. This is like right before you get to pyruvate, phosphoenol pyruvate. This is the product of step 9 of glycolysis, and this is a negative inhibitor of phosphofructokinase. Okay, so step 3 here is catalyzed by PFK. And the logic of this sort of makes sense. If you have tons of product down here, then you don't need the pathway to still be going all gung-ho, right? You can have it slow down and it'll conserve cellular resources, okay? Whereas if you have a decrease here, then that inhibition will be released and then the enzyme can push forward. And so that's an example of an allosteric feedback mechanism, and this would be negative feedback. Sometimes you'll have things that are positive feedback mechanisms. An example would be oxytocin, right? When oxytocin acts on its effectors, it will upregulate oxytocin, so it's a positive feedback mechanism. And what we're seeing over here is the differeces in the types of curves. So this should look very familiar to you in terms of how myoglobin and hemoglobin worked, right? Here's what we have when we look at no regulation versus allosteric regulation of phosphofructokinase. And then just to finish up, mechanisms for regulating enzyme activity. So how it would need to be done. There's a ton of different ways. We've sort of alluded to some of them already. Rates of enzyme synthesis and degradation is definitely a major form. So also, in terms of transcription and translation, but also, synthesis of zymogens as opposed to a fully functional enzyme. Things like that will affect the activity. A second way that it could be regulated would be the location of the enzyme. So eukaryotic cells, of course, as we've mentioned, have these membrane-bound locations within the cells, and you may want to, just have an enzyme be working, just in a lysosome or just in the nucleus, and not just within cellular compartments, but even in different cells of an organism. Some cells may have an enzyme being activated versus in another part of the organism. The third point here is a chemical signal may be required in order to activate the enzyme. So a common one would be a calcium burst that may come from the endoplasmic reticulum. So your calcium ions that are being released are going to activate, or would be required to activate, the enzyme. And then finally, there could be covalent modifications to an enzyme. Sometimes phosphorylation of an enzyme, for example, would be required before it is active. And so you can have kinases, and this can work in cascades, right? You can have kinases and phosphatases that will be involved in the process. So it's just like a little overview of regulation. Okay, so hopefully that has not clouded things up too much, in particular with respect to non-competitive inhibition, but it is certainly more accurate. Okay.